Hello, welcome. Let's talk about Chancelloриids. Wait till the model spins around. We're talking about the orange thing up there, and I think the other orange things as well, possibly, but definitely the spiky little orange thing. The rest of the things in the picture are sponges. That's a clue. And remember, sponges and Chancelloриids, these are all animals, even though they're sitting still their entire life and covered in thorns like a blackberry bush. We got to interpret these things as animals because from the sediments that they are in, they are living very deep down, which would be comparable to far too deep for there to be much oxygen or much sunlight, like enough sunlight for actual photosynthesis. They live farther down than modern plants and algae can comfortably live now, which is how we know they're not plants or algae. Part of it, yeah. The rest is that they're different. They're not plants. All right, so let's go back up to the top, get our nice little splash screen, and yeah, let's talk Chancelloриids. I like Chancelloриids. I find them fascinating because just look at them. It's just like a little hollow, like a pear, upside down pear, attached by its stem, covered in thorns. How fantastic is that? These are the original ones. These are called Chancelloria pentacta, and they come from Utah, and they are fantastic, and they're actually fairly expensive. I have not been able to afford even a broken one of these yet, although I think I just got to go out there, and then there'll probably be one. So, looking a little more closely at the structure of these things, what are they? Um, so you'll see that each of these things, so it has a sort of a pointy end at the bottom, and that's where it's usually going to be attached to the substrate. And sometimes you'll find these things in clumps with stuff like sponges, which is exactly what's going on up here. This is based on a particular fossil with all these different things living together. So it's a community of things that were able to survive in the same conditions. And we know about a lot of sponges that live very, very deep in the ocean. Think of some of those glass sponges, hexactinellids, with the really intricate little skeletons. And those things are found as far down as you find life. I think it's more that they're limited by the availability of carbon sources than anything else. So, looking at the organism itself, all right, what are the parts that we've got to work with here? This little animal mystery. So, we've got a little tuft of spines right up here. And you see those are at the opposite end to the end that's attached. So, it's like there's something that's going on up there, and it's got a little, spiky hairdo on the top. That's neat. Also, on this particular fossil, there is a concentration of pyrite at this spot. Now, this is a fossil that's replaced by pyrite, and so it doesn't mean that it was originally made of fool's gold, which would have been awesome. Yeah. It means that there's maybe some extra iron there, or iron loving bacteria deposited themselves there for some reason. I don't think we know fully, or at least I don't understand fully from reading the papers, why that is the case, but that is the case. And you'll see the same thing over here, pretty much. And if you take these things, you can actually x-ray them because they're made of metal, and the rock around them is very low in metal, and so they will pop right out if you do that. You can put them, float them under xylene, I think is one of the ways they take these pictures. And yeah, they look gorgeous. Look at that. Yeah, like a little three-dimensional thing. So we first learned about Chancelloриids, or rather our first evidence of Chancelloриids are sclerites in small shelly fauna. Small shelly fauna come from right at the bottom of the Cambrian. Maybe, possibly, some of it is from the top of the Ediacaran. So these come from times and places where we don't get good body fossils, but we get broken up remnants of all the stuff that had hard parts that was living there at the spot. So it's like, well, it's like a bunch of puzzles. It's not like a puzzle. It's like all the puzzles, and most of the pieces are missing. And good luck to you, boss. Yeah. But you get all these little things in there. You get little tubes. You get little bananas in there that kind of look like claws, and maybe claws. You get, I don't know what that is. It looks like a 4M maybe. And you get these little things. They've got like a central boss, which sometimes you can see that's actually a spine in some of these. And in some of them, here, the spine is going off into the screen like that. Kind of like a caltrop, those things that you put out so that horses step on them and get lame. Or I think even those are kind of mythological. But yeah, this is very much the biological equivalent of that, and they are very real. Another interesting thing that you can't quite see here, but you can maybe see on the side of this one. So this is 2, 3, 4, 5. All of these things are the same as this, or rather they're the same type of thing, but they're clearly a different version. So think maybe a different genus in the same family, something like that. And that's how they're currently interpreted, I think. Yeah, they're at least all Chancelloriid parts. That much I know for sure. Yeah, they're hollow. So look at that. It's like a crispy coating on the outside and then hollow inside. That can have a couple meanings. It can mean that there was never anything inside and they formed around something. Like if there's a cell inside, it can develop a shell around itself. You cut open a bone from a dinosaur or a human or a turtle or whatever, and you will find these little trapped, dead, or dying, nuclei in there. They build the bone and they build it all around themselves, but eventually they cover themselves over completely and they're just left there. And eventually, when you get a fossil, you just have a little hole that's the right shape for essentially the nucleus and a little tiny bit of cytoplasm. And that's all there was of these cells. Yeah, all that's left. So, small shelly fauna, very interesting. And of course, we have a sampling of small shelly fauna. You would normally need to digest these things in some kind of acid. And you got to be careful with the acid, like get the right type that will leave the fossils alone but get rid of some of the matrix. I haven't tried to mess with our sample yet, so it just looks like a gray rock, but it's from the right time and place and formation where small shelly fauna are routinely found. And you have to process a decent amount of rock to get a nice collection like this. That's probably a couple pounds worth of rock, maybe more, yeah. And sometimes you have to soak these in acid for months or like a year, and so it's not very conducive to doing a thing during a course. Now is it? Yeah, unfortunately. Okay, so what else can we figure out about these? So they're covered in thorns, covered in spines, and so there'll be spines that stick up and kind of run along the side of the thing. They may have stuck out a little bit. And there are spines, like here's one, and these would be sticking straight out, so like toward you, the viewer. And so those would definitely be, stay back or don't eat me or, just get off my lawn kind of spines. But then underneath the spines, look at this sort of leathery texture that you've got there. You can see maybe a little bit of it up here. This is the intact skin. And the intact skin is actually, it looks fairly watertight. So, like, all right, well, it's a thing that's in the water and it's just, it's like got a skin on it, like an animal, like, or like an apple, yeah. All right, let's look a little closer. So here is the opening at the top. And in some of these that are very nicely pyritized, like turned into fool's gold, you can see what it is if they get caught at just the right angle, if they get sort of squished like this way instead of falling over and getting squished this way, which is what we were looking at before. And you can see that there's a little hole and it's got long spines that are poking into the hole. So there's one big hole at the top of the animal and a whole bunch of spines to keep things off the sides and a whole ring of spines that would maybe let something like water pass through and would keep something like, I don't know, trilobites or fish or shrimp or you name it out. And because a thing looks like that doesn't mean that's what it does, but it's a piece of data that we have to put into our understanding of what this creature is. Next piece of data comes from this one, Allonnia nuda, which is just another Chancelloriid. It has very few little spine clusters. So is it living in an area where there are fewer predators and it just lost the ability to make spines and it didn't matter? Maybe, I don't know. It's not older than the ones that have spines. It lives at the same time as those. In roughly the same place, it's found in the same formation, apparently, but maybe in a different part of the formation. That evidence is usually not as clear in the papers. But what you can see on this is that, yeah, you got that nice sort of skin texture going on up here, and you got the nice spines here. They get sort of compressed into the body a little bit when it's, because remember, this thing has died, fallen over, and had a whole bunch of mud and then dirt and then rock sitting on top of it. And so it gets flattened out real flat. And yeah, that just shows this was a thing that could be flattened. And it doesn't look like it had any hard parts except for the spikes, these little guys. But you look on this and you can see that the smooth part is in places covered with these little circular, look at those little donuts. Yeah. And those we think are pores. So if those are little pores and they're all in the side of the creature and there's one big pore in the top center of the creature, then what kind of creature are we talking about? There's only one thing that you know that satisfies those criteria. It's a sponge, right? So again, easy to say it is sponge-like, and for a long time in the literature that is exactly what people have said, and some people still do say. It's a sponge-grade organism, sponge-like organism, because you probably need DNA to know for sure if it's really a sponge, because it's not, there are some things that are different. Yeah. So here's a zoomed in part of this particular Allonnia, and look at that. Even though it's almost completely missing spines, up near the tuft it has all kinds of spines, and it still has that little circlet of long straight spines that don't look like the other ones. So you're thinking now there's got to be some different genetic program. Like when the spines are forming, they have to know where they are on the organism, and they have, so they can tell whether to make like no spines or a bunch of spines. And then if they are around this hole, they have to throw out the original game plan, which is to make a spine with usually 4 little prongs coming out of it, one of which is pointed either straight up or straight down in this image. And instead go with the long skinny spine with nothing sticking out the sides. And they do that. So you've got something like the genes that control how many fingers our babies develop with, and how many toes and how many arms and how many legs. You've got some kind of global control over body morphology, like as it grows. So yeah, that's interesting. That's yeah, interesting and somewhat revealing. And also, if this is something like a sponge, then all the water is going to be coming out of this central hole. So any organism that wanted to leach off the sponge could go into that hole, and it could just sort of burrow into the sponge material. You know, if like the skin is fairly tough but the inside is not, that would be a way for parasites to get in. But also, if you're not talking about a real parasite, just something that wants a free ride, it could just clamp itself down over that hole. And then anything that goes through the sponge but doesn't get captured, which is frankly going to be a lot of the particles, is going to go right into the mouth of this other thing. And this other thing won't even have to generate any water current. The sponge will just keep feeding it. It'll be an endless buffet coming straight at you, courtesy of the sponge. And that's probably not great for the sponge, although it may not suffer too much, but definitely great for the parasite. And it looks as though these sponges, if we're interpreting the features correctly, are set up to prevent that. So we can infer that this may have been a problem, and this may be the solution that was selected through natural selection. Yeah. Now, what we know at this point is that there are actually groups of sponges, and these sponges look a lot like Chancelloriids. They've got these little rings of spines around the top. This is one particular one, Sicon ciliatum, and I forget what they're staining here. They're staining the living tissue, yeah. And so that's what the green stuff is. So you can see most of it is just empty space that's going to be filled with water. And you can think if this thing flipped over or tipped over and then got sort of compressed, you could just squeeze the water out of that and it would squeeze down to flatness in no time at all. It's just like an empty shopping bag, something like that. But the idea is that this thing shows similar features to what we get on Chancelloриids, so it's plausible that Chancelloриids are like sponges. One problem with that is that modern sponges do not have hollow spicules. They actually make the spicules. There are cells that secrete them, and manufacture them, and none of those are hollow. But Chancellorid spicules are. If spicules is the right word for them, that's just what we call them. Okay, so let's go to Mother Russia for a minute, shall we? Let's. All right. So this is in Cambrian times, stage 4, and this is over in the eastern part of Russia, like Siberia, basically, is what you would call this. And so they're showing the division in time, and then they're showing a little box around the area where they're hunting. This is a standard sort of figure 1 for any geology paper or paleontology paper. They got to show you where they got the stuff, and then they'll usually show you the order of the different layers. And so here you can see a lovely order of the layers diagram. And usually these solid black things end up being black shales. The little sort of bricks are usually shorthand for limestone. It's usually little dots when they want to show sandstone. And there'll be a key over here anyway. There you go. So calcareous shale is this sort of liney thing. And there you go. So, and then they'll show you in this where they found the fossils. So this is usually figure 2. In this case, they combine them both to make a great big figure 1, and good on them. Yeah, that's a great way to do it. Interesting thing, they show several very early sponges from the Cambrian here, and these are things that are not Chancelloриids at all, and they are pretty non-controversially sponges. They look like sponges. They seem to have a lot of sponge-like features. Here you can see the spicules. Look at all of those laying down there. Yeah, looking good. If you zoom in, so here's one of the big spicules. There are these little fibers under them. They're like a smaller version of spicules. Now, most sponges today have a substance called spongin, which is like, they're making spicules out of it, but they're like bendy, somewhat flexible spicules. This is why using a bath sponge is not an excruciating, like sandpapering yourself. Because they're a little bit bendy, and that's why bath sponges work as bath sponges. But if you look at them, notice they're hollow. Yeah, the spicules on these particular early sponges are hollow, and these are very securely sponges. Let's look at another one. So here you go. Here's another one, and look at that. Hollow, hollow, and all these little things, hollow. So yeah, there's different sizes of them, but it would appear that, at least in the early sponges, they either were hollow or they fossilized hollow. And probably it's the latter of those two, because in fossilization, you're going to lose a lot, especially a lot of that soft tissue. You'll just get an impression of it at best. But you'll lose a lot of the stuff like organic components, like carbohydrates and protein and stuff like that. And spongin is a protein. That's the thing that demospongia, that's the sort of common sponges, the most abundant sponges. That's what they make their skeleton out of, for the most part. They're demosponges that make other kinds of skeletons, because sponges are very interesting or kind of a mess, depending on your perspective. I think they're wonderful. Yeah, just look at them. Look at them rotating up there. All right, so from this paper, they actually found a few things, and so they did some analysis in particular on the way the bodies are put together, the way the spicules look, whether the spicules are branched or hollow or straight. And you can put these things into a computer the same as you would put in DNA letters or protein letters, and you can make a family tree. And so we have to assume they've done a good job on that tree, because we're not at a position where we can analyze all of that, or where we have time to analyze that in enough detail right now. But what they find is that, so these things, they say that hexactinellids, the glass sponges, are probably an earlier branch. And one can only assume that Chancelloриids are somewhere around here. But a couple of the new sponges they found are right here and here. And so they're showing you what's going on here. So the organic fiber skeleton, they're saying, is what they develop at point 1. And so they're saying maybe these fibers were originally organic, something like protein carbohydrate mix, glycoprotein, which would make a lot of sense in terms of modern sponges. And they've just fossilized to look as though they are hollow, or maybe they had a core of something like protein and that went away on fossilization, and so you're only left with the hard parts that were laid down outside of that, which is also a thing that could happen. Doesn't have a lot of precedent in modern sponges that I'm aware of, but why should modern sponges reproduce every feature of their ancestors? I mean, they've had to compete with, and they have outcompeted these ancestors. So it may be there are better ways to do things, or it may be that the randomness that is inherent in evolution gave us new ways to do things. So there you go. Chancelloriids. Probably sponges, definitely amazing, very spiky, wouldn't want to step on one, especially because you'd have to be down pretty deep in the ocean and there's probably no oxygen down there and it would probably crush you. Yeah. So don't don't do that. Be nice to the Chancelloriids. Yeah, and so these are going to be some review questions to make sure you know what's going on with Chancelloriids. So we have lots and lots of Chancelloriids because I find them wonderful, and so I bought a bunch of fossils of these things and some other early sponges, and you can compare and contrast. That's what we'll do. Thanks.